I'm ready.
My name's Jenna DeCata, and I am majoring in biology, specifically in genetics. I'm also getting a minor in Latin, and I am a junior. So we all know that things like pollution, radiation, smoking, and even UV ray damage or sunburn can cause things like DNA damage and mutations. But even if you avoid these things your whole life, your body and your DNA will still undergo oxidative damage due to processes like metabolism. And luckily, humans have evolved to have a repair mechanism built into their body. The bad news is that this repair mechanism doesn't always work, which is why I'm studying a mechanism called BIR, or break-induced replication, which repairs double-stranded breaks. And why I'm studying BIR specifically is because it's linked to chromosomal rearrangements. And these chromosomal rearrangements can also be linked to causing genetic disorders. But in order to better understand this mechanism, we need to look at the proteins. And there are many proteins in BIR alone, but I am specifically looking at who is doing what in this mechanism, but specifically a gene that produces a protein called SRS2. And I'm interested in SRS2 for two reasons. First of all, SRS2 is a helicase protein. And as you can see here, the helicase opens the DNA so that things like repair, replication, or other mechanisms can happen. The other reason that I'm looking at SRS2 is because it's been studied if this gene is completely removed from the cell and a break is induced, the cell is not able to repair itself completely and the chromosome degrades, as you can see here, as opposed to the wild type, which is able to completely repair itself as time goes on. This interesting characteristic, though, hasn't really been linked to a certain part of the SRS gene, which is where my research starts. I'm using a genetic approach to look at cell viability, the type of repair that's going on, and the DNA products of this attempt at repair, using 9 different yeast strains, all with one mutation in one of these 3 regions, to examine the role and the effect of this gene on BIR and determine why it is so necessary for DNA repair. Thank you.